Hello students, in previous part we have discussed about male reproductive system and female reproductive system. In this part we discuss on gametogenesis. Gametogenesis. Formation of gamete is called gametogenesis. And gametes in males are sperms. In males gametes are sperm and female gametes are ovum. Formation of sperm is called spermatogenesis. It means in male sperm are produced and this is called spermatogenesis. Before going to this topic, we have to discuss some different animals. We can categorize the animals on the basis of intra-abdominal testis and extra-abdominal testis. The animals we can categorize into three groups. The first group is intra-abdominal testis. The animals in which contain testes all the lifetime in the abdominal. The testes don't comes into the extra, or you can say that just in the scrotal sac. The scrotal sac remains in the abdominal cavity. Testes remains in the abdominal cavity. It means the testes are able to produce sperm at the body temperature. Those animals are elephant and whales. They have the ability to produce sperm at the body temperature. So the testes remains inside the abdominal cavity. So they can be categorized into intra-abdominal testis animals. The second type of animals are extra-abdominal testis. Just like as human being horse, these organisms, the testes descend into the scrotal cell outside the abdominal cavity, and all the lifetime testes remains here. In human being, horse, the testes remains outside the abdominal cavity. In the scrotal sac. Because in these organisms, testes cannot be able to produce sperm at the body temperature. So testes descend. Testes are produced in abdominal cavity, but they descend into the scrotal sac so that they can able to produce sperm. And third types of organisms are seasonal breeders. Some organisms such like as llama, bats and dogs, these organisms only able to reproduce at the seasons. At some seasons, the season time may be months and days. So some organisms are seasonal breeders. Seasonal breeders. During the season's time, breeding season, the testes descend into the scrotal sac from the abdominal cavity. As the seasons off, the testes ascend again into the abdominal cavity. Dogs, llama is the species of camel in South America. Bats, the testes descend in the scrotal sac. Sac outside the body. Yeah, outside the abdominal cavity. During breeding season. Breeding season. And as the breeding season of testes again ascend into the abdominal cavity from scrotus. After breeding season, the testes ascend in the abdominal cavity from scrotum. It means these organ animals are able to reproduce only breeding seasons. At this breeding season, the testes descend from abdominal cavity to the scrotum, and as the seasonal breeding season of testes again ascend into the abdominal cavity. First of all, we categorize this type. The next one is spermatogenesis. What is spermatogenesis? Formation of sperm is called spermatogenesis. Again, spermatogenesis. Formation of sperm is called spermatogenesis. The spermatogenesis process is completed in two phases. This phase, formation of spermatogenesis as sperm, is completed in two phases. One phase, first phase is formation of spermatid. Spermatid is a circular non-motile cell. Formation of spermatid, and second is formation of sperm. Spermatid is non-motile, but sperms are motile. Formation of sperm from non-motile spermatid. So it is called spermiogenesis. So we can say that there are two phases occur during the spermatogenesis. First is formation of spermatid, and second one is spermiogenesis. First we will discuss all these two phases. The formation of spermatid is completed in three stages. Spermatogenesis is the process in which sperms are produced. The spermatogenesis process completed in two phases. First phase is formation of spermatid, and second phase is spermiogenesis. Spermatid completed in three stages, and those three phases are: first is multiplication phase. The second phase is growth phase, and third phase is maturation phase. How these phases can be occurred? First of all, discuss on formation of spermatid. We know that sperms are produced in the seminiferous tubules. This is the epidermis layer of seminiferous tubules. Epidermis of seminiferous tubules. You know, the sperms are produced in testes with seminiferous tubules. At the epidermis of seminiferous tubules, there are two types of cells. Some cuboidal cells, mostly cuboidal cells, which are known as germinal cells, and there are some conical shaped cells. The conical shaped cells are called sertolins. This is the Sertoli cell, and the cuboidal cell is germinal cell. How is the process of formation of sperm? The first process, we know that sperms are produced in seminiferous tubules. At the epidermis of seminiferous tubule, there are two types of cell: germinal cell and Sertoli cell. Some of germinal cell, as it gain maturity, this germinal cell enters into the mitotic phase. Mitotic phase. First of all, germinal cell is a diploid cell. As a diploid cell, the diploid cell enters into the mitotic phase, then it changes into two diploid cell. And these two again divided and change into four diploid cells. Formation of germinal cell to spermatogonia. This process may occur during the mitotic phase. As the germinal cell enters into the mitotic phase, first of all, the germinal cell is a diploid cell. In mitotic phase, it is the equational phase. The number of chromosomes remains same. This diploid germinal cell divided into the four diploid cells. These are called spermatogonia. Spermatogonia. Out of these spermatogonia, some cells continue take part in the mitotic phase. Suppose these are the cells. We categorize these cells into two parts: X type and second one is Y type. How we can categorize differentiate? How we can differentiate? Some cells continue remains in the mitotic phase. It means increase the number of cells. Increase the number of cells, that is called multiplication phase. This phase occurred in multiplication phase. In multiplication phase, germinal cell increase their number. During multiplication phase or mitotic phase, some cell remains continue into the mitotic division. They continue increase their number and remains into the mitotic division. These cells are called stem cells. What do you understand by stem cells? The germinal cell which increase their number due to the mitotic phase, due to the mitotic division. These cells continue remains into the mitotic divisions and they increase their continuously numbers. These cells are called stem cells. And some cells, suppose X type cell remains into the mitotic division, so they are called stem cells. The another Y type cell, they enter into the another phase. The another phase is growth phase. These cells, due to the accumulation of nutrition provided by the Sertoli cell, the Y type cell then enters, they leave the mitotic division, they enter another phase. They start to grow, they increase their size due to the accumulation of nutrition. This cell start to grow, and as the cell increase their size, that phase is called growth phase. This is called growth phase. Growth phase, as the cell gain growth, increase their size, this is called primary spermatocytes. Spermatocytes. Again listen, the cell which doesn't enter, the cells which do not enter into the mitotic division, they start to grow their size, due to the accumulation of food, nutrition. The cell which increase their size, that enters into the growth phase. The cell which gain growth, that is called primary spermatocyte. And primary spermatocyte is a diploid cell because there is no division can take place here. Then this cell enters into the M phase, means meiosis division, not mitotic, meiosis. meiosis division. And we know that meiosis is of two types, M1 phase and M2 phase. M1 phase is reductional phase. So the diploid cell as it enters into the meiosis division M1 phase, they produce two haploid cells, that is also haploid and haploid. From a diploid primary spermatocyte cell as it enters into the meiosis M1 phase, the number of chromosomes remains half, and these cells are haploid cells. The cells which are produced from the diploid primary spermatocyte, these are called secondary spermatocyte. These secondary spermatocytes are haploid cells. Again, these cells enter into the M2 phase. M2 phase, there are formation of 4 cells. These are also haploid. These cells are called spermatid. How is this formation of spermatid can take place? We have seen that the formation of spermatid completed in 3 phases, but we have discussed only 2 phases. First phase, multiplication phase. Epidermis layer of seminiferous tubules, it contains two types of cells. First is germinal cell, second one is sertoli cell. The germinal cell, that is diploid, enters into the mitotic division. As it enters into the mitotic division, it produces 2 diploid cells, then 2 diploid to 4 diploid cells. These 4 diploid cells are called spermatogonia. As the spermatogonia form, we categorize these cells into 2 categories. Some cell remains continue into the mitotic division, increase their number, so that is called multiplication phase. And the cell which continue remains into the mitotic division, those are called stem cells. Those are X type cells. The second Y type cell, they leave the mitotic division. They enter another phase. Due to the accumulation of food, the cell gain food and increase their size. And this is called growth phase. This is the second phase. First phase. The growth, as the cells gain its growth, this is called primary spermatocyte. The primary spermatocyte is diploid cell. Then this primary spermatocyte enters into the meiosis division, not mitotic, meiosis division. And meiosis first is reductional phase. So a diploid cell change into 2 haploid cells. The 2 haploid cells which form from the primary spermatocyte, those are called secondary spermatocytes. These are haploid. And then the meiosis 2, second phase, then these cells enters into the meiosis 2. Meiosis 2 is an equational phase. In which the cell new are produced, that is similar to this one. So it produces 4 haploids. The cell which produce from secondary spermatocytes, those are called spermatid. So what are the spermatids? Spermatid is a circular non-motile haploid cell. Spermatid is a circular non-motile haploid cell. The first phase is complete. Then the phase enters into the second phase. Second phase is spermatogenesis. Spermatogenesis means formation of motile sperm from non-motile circular cell. It means non-motile spermatid. Formation of motile sperm from non-motile spermatid. This is called spermatogenesis. For example, these are the spermatid and they produce motile spermatid. It means formation of motile sperm from non-motile spermatid, that is called spermatogenesis. This is the second phase of it. As the second phase occurs, there are some changes can be seen. It means the sperms as produced from spermatid, there are some changes can be seen in the spermatid. What are the changes occurred? Those changes are changes occurred or changes can be seen during formation of sperm. Formation of sperm from spermatid. The first, condensation of nucleus. The nucleus condense and reduce RNA with acidic protein. So it remains only basic protein with the DNA. Condensation of nucleus. During the condensation of nucleus, it loses RNA and acidic protein. So in the nucleus only remains DNA and basic protein. These are reduced. During the condensation of nucleus, RNA, mRNA, messenger RNA and acidic protein is reduced. So the nucleus contains only DNA and basic protein. The second phase is nucleus change into pointed end, flat or oval. Nucleus change into pointed and oval shape or flat shape. This is the second process, second change occurred during the formation of sperm from spermatid. The next is Golgi body. Golgi body converts into two types, or we can say that the Golgi body change into two types. One main Golgi body which change into acrosome that contains sperm lysine, and second degenerate that is called Golgi rest. Golgi body. First of all, Golgi body converts into two types. Change into two types. First, the main converts into acrosome. Acrosome which contains sperm lysine. Sperm lysine an enzyme which diffuse the corona radiata of ovum in female body. And second, the small part of Golgi body which degenerate, that is called Golgi rest. This is the third change. Fourth change occurred during this phase. The mitochondria which is found in middle piece of sperm, that is a ribbon shape or spiral shape. That ribbon shape or spiral shape mitochondria is called neighborhoods. Mitochondria found in the middle piece of sperm in spiral or ribbon shape. The spiral or ribbon shape mitochondria is called Neberkern. What is Neberkern? Neberkern, the mitochondria which is found in middle piece of sperm in spiral shape or ribbon like, that's called Neberkern. The next change occurred. The sperms is covered with plasma membrane. Sperm is covered with plasma membrane. The whole body of sperm is covered with plasma membrane. And last change occurred, cytoplasm. Cytoplasm present only in tail part of the sperm, not whole body. Cytoplasm present only in tail of sperm, not the whole body. This whole process is known as spermatogenesis. What is spermatogenesis? Formation of sperm is called spermatogenesis. Then we have seen spermiogenesis. What do you mean by spermiogenesis? Formation of sperm means motile sperm from non-motile spermatid, that is called spermiogenesis. Listen, one more term is spermiation. Spermiation, as we have seen that this is the epidermis of seminiferous tubules, and at seminiferous tubules there are two types of cells, ovoidal germinal cell and conical sertoli cell. These cells germinal cell produce sperms, and these sperms remain attached with the sertoli cell. This is the sertoli cell. The sperm attached with the sertoli cell. These are the sperms. As the sperm mature, it means mature sperm are released from sertoli cell. As the sperm mature, these mature sperms are released through the sertoli cell and comes into the seminiferous tubules, lumen of seminiferous tubules, and then they pass through the column retestis, then the vasa afferentia, vasa deferens, and ejaculation. This release of mature sperm from sertoli cell, this is called spermiation. So we have discussed about three main processes: spermatogenesis, formation of sperm is called spermatogenesis. Second phase is spermiogenesis. It means formation of motile sperm from non-motile spermatid. That is called spermiogenesis. And third, the related term is spermiation, release of mature sperm from sertoli cell. This process is called spermiation. In next part we will discuss on the male sperm structure.